Our first speaker, Dr. Benjamin Martin from Harvard Medical School. And today he'll be talking to us about global identification of the switch SNF targets reveals compensation by EP4. Take it away, Martin.
Awesome. Thank you. Thank you so much. Let me just share my screen. All right. You should see my slides. So hello, everyone. First, I just want to thank the CSMB and the organizing team for putting the seminar series together. I think it's a great thing you're doing, and I really appreciate the opportunity to share my work with you all today. The story is one where we've uncovered a surprising compensation mechanism with a remodeler called EP400 compensating for loss of mammalian switch SNF. It's a story rooted in basic science, but with some broad implications for cancer biology. But first, let me start with the big picture. So as we all know, our genetic information is stored in DNA. And for this info to be functional, DNA must be read and converted into RNA, a process called transcription. While all our cells have the same DNA, this information is used differently depending on the biological context. For example, some genes are expressed in all cell types, or a gene such as gene B shown here could be expressed only in a single cell type, leading to different cellular functions. And in today's seminar, I'll be focusing on this first step of transcription, which is really important not only for proper development, but also as alterations in RNA synthesis contribute to numerous disease states. And so we're interested in how transcription is regulated in mammalian cells. Okay, there we go. The main focus of my talk today will be chromatin remodelers and the mammalian switch SNF complex in particular. This complex, like all chromatin remodelers, has an ATPase that can move and evict nucleosomes from DNA. And for switch SNF, this is provided by the BRG1 protein. And its remodeling activity is really important to allow the machineries that carry out DNA replication, repair, or transcription to access DNA. And we're particularly interested in this complex because, as many have shown, it's so frequently mutated in cancer and other diseases. And our motivation for the study was the observation made by numerous groups that disruption of switch SNF resulted in rather modest effects on gene expression, despite it being such a major remodeler and co-activator, suggesting a compensation mechanism. And I'll circle back to this point after giving an introduction on its role in gene activation. So in eukaryotes, DNA is packaged by histones into the nucleosome, and this forms a barrier to transcription initiation. So for gene activation, one of the first steps starts with transcription factors binding and chromatin remodeling by complexes like switch SNF sliding or evicting nucleosomes away from the promoter. This allows for the pre-initiation complex to form, leading to transcription initiation and promoter escape. Shortly after promoter escape, Pol2 then pauses before regulated pause release and progression into productive elongation, where the polymerase must overcome nucleosomal barriers, sometimes with the aid of chromatin remodelers. Along with promoters, another crucial DNA element controlling gene activation is the enhancer. These are short regions of DNA that can act at a distance, sometimes a few kb and up to a megabase, to stimulate transcription from gene promoters. And enhancers are thought to be one of the major mechanisms for regulating gene expression in metazoans. Okay. And I'll note that remodeler activity is generally important for all active regulatory elements, with remodelers generating accessible chromatin at both promoters and enhancers. And indeed, enhancers share many features of promoters, including accessibility, transcription factor binding, histone modifications, and of course, transcription by Pol2. Now let's look at this complex in more detail. As I said, it's a major remodeler and co-activator that's known to open up chromatin for transcription factor binding at many loci across the genome. And as I noted before, it's frequently mutated in cancer. And as I already alluded to, we got particularly interested in this complex because of a real conundrum in the field. Although everyone agrees it's really important and plays a broad role in generating accessible chromatin, many groups have found that disruption of switch SNF activity by targeting the catalytic subunits and inactivating all the different flavors of switch SNF complex, these perturbations typically result in selective rather than global alterations in gene expression. And we decided the time was right to probe this question because of the development of a fast-acting and specific inhibitor of BRG1 and BRM. With this inhibitor, some previous work, beautiful studies have shown that treatment of mouse or human cells causes a rapid and broad loss of accessibility and transcription factor binding at putative enhancers. And these findings were recapitulated by rapid depletion using digrons and protac degraders of BRG1 and BRM, BRM being the paralog of BRG1. However, these work raise an important question. which is how can a widespread reduction in accessibility at regulatory elements only cause modest or specific changes in gene expression? And this will be the focus of my talk today. To address this question, we've used the catalytic inhibitor BRM14 to probe the direct effects of switch-NIF inhibition. And I'll note that I performed these experiments together with Eileen Blondie, a talented student recently graduated from the Edelman lab. And this work just recently was published a few weeks ago. But first, let me start by explaining to you the two key methods we have used in this study. So first, ATAC-seq. This is a technique that uses a transposase to insert sequencing adapters into the genome. Now, the transposase used, this TN5, is occluded by nucleosomes, meaning that primarily inserts the sequencing adapters into nucleosome-free regions of DNA. We can then use the inserted adapters to sequence the DNA within, and thus have a powerful assay to profile open or accessible chromatin. So what does this actually look like? Here is a ATAC-seq signal around an example gene. And you see a characteristic peak of a ATAC signal at the promoter. And we can also see a peak upstream of the gene that may be a regulatory element. The second method to tell you about is precision run-on sequencing, or PRO-seq. For this method, collected cells are permealized, which means that all of the endogenous NTPs are washed away. Then we perform a run-on assay with biotinylated NTPs. And because the biotin is very bulky, Pol2 can really only add one nucleotide before arresting. Then we can use the biotin to purify the nascent RNAs and ligate on sequencing adapters, which allow us to sequence the 3' end of the RNA. This then gives us the position of the active site for all of the elongation competent RNA polymerases at single nucleotide resolution. So PRO-seq is a real favorite in the lab as it allows us to powerfully profile transcription at both enhancers and genes with great sensitivity and at high resolution. So what does PRO-seq data look like? If we layer in our PRO-seq data at our example gene, we now see where active RNA polymerases are. Note that PRO-seq gives us strand-specific data. So we have RNA Pol2 positions on both the forward and the reverse strands. And we see a characteristically big peak at the gene start on the reverse strand, which is the pause polymerase. And we also see some transcription activity at the upstream ATAC-seq peak. Now, as a basis for our study, we use chromatin accessibility and nascent transcription to define active regulatory elements. And we did this using a fairly simple approach where peaks of PRO-seq signal that were near annotated genes were called promoters, shown here in green. And transcribed regions that were distal from any annotated promoters were considered putative enhancers, seen here in purple. And these loci were confirmed using ATAC-seq, so the sites for investigating are both accessible and transcriptionally active. And through this method, we identified over 13,000 active promoters and over 32,000 active enhancers in our mouse embryonic stem cells. And for switch-SNF and ES cells, BRG1 is the sole ATPase expressed. And as you can see here in ChIP-seq for BRG1, which is a method for mapping protein binding across the genome, BRG1 associates both promoter and enhancer regions. We've got everything set to now ask what happens when we inhibit switch-SNF. So we treated MESCs with the inhibitor BRM14 for 2 hours and performed ATAC-seq. Here I'm showing the data as a heat map with each row representing an enhancer and with black indicating chromatin accessibility. And consistent with previous work, when we look at all of our enhancers in a heat map, in control cells, we see accessibility by ATAC-seq at these loci. And after 2 hours of switch-SNF inhibitor, we see less ATAC-seq signal nearly everywhere, meaning that all enhancers become less accessible. Now, to make this easier to visualize, we can subtract the control from the inhibitor-treated sample and show this as a color scale, where blue means loss of accessibility and red means gain. And we can see that there's mostly blue in this heat map, meaning less accessibility. And if we look at BRG1 ChIP-seq, we can see that the enhancers with the biggest decrease in accessibility at the top of the figure, they are nicely enhanced with the most BRG1 bound. So overall, these data confirm that switch-SNF functions universally to open up chromatin, and it does so at nearly all enhancers. And if we extend this inhibition to 4 and to 8 hours, the results don't change much, which you can see by the predominantly blue signal on the heat map all the way through 8 hours of switch-SNF inhibition. And complementary work from the Schubler and Kubicex labs have confirmed, as you expect, that this loss of accessibility causes a prolonged loss of transcription factor binding. Now, if we look at enhancer RNA synthesis using PRO-seq over this time course, you can see that consistent with a loss of transcription factors, we see a loss of transcription activity. So switch-SNF inhibition shuts down most enhancers at the level of chromatin accessibility, transcription factor binding, and eRNA synthesis. Now, this is both kind of expected, but also surprising, because this result still leaves this question open. Namely, how does a global decrease in enhancer accessibility ultimately lead to only selective effects on gene expression? So we next want to look at what happened at gene promoters. And so to compare apples to apples, we looked at our attack-seq and pro-seq during the time course of BRM14 treatment and found at 2 hours, a loss of accessibility with a corresponding loss of transcription initiation. So at this time point, promoters look similar to enhancers. But now this changes as we extend the duration of treatment to 4 or to 8 hours, when a majority of promoters bounce right back and even overcompensate in accessibility, with a red signal in the heat map indicating attack-seq signal increasing relative to DMSO controls. And when accessibility bounces back, transcription follows. So when we investigate gene activity at the 4 or the 8 hour time point, most promoters are now back to their normal expression levels. So this suggests that cells compensate for loss of switch SNF activity at the majority of promoters. And this means that after 4 or 8 hours of switch SNF inhibition, what we're really studying at promoters is variable recovery and compensation. To investigate these different dynamics more rigorously, we clustered promoters using the relative attack-seq levels at each time point. Our analysis identified 4 clusters whose accessibility was altered by BRM14 different kinetic behaviors. Importantly, whereas most promoters are capable of rapidly reinstating chromatin accessibility, cluster 1 promoters had reduced accessibility over the time course of treatment. And this cluster is enriched in genes involved in development, differentiation, and cell type specific expression. And in data that I don't have time to show, we found that this recovery is really disconnected from what is happening at nearby enhancers, which exhibit sustained repression through the time course, pointing to a promoter autonomous compensation and recovery for loss of switch SNF activity. So we next asked what makes certain promoters so susceptible to loss of switch SNF activity. And to do this, we first looked at features of these promoters in control cells. Looking at chromatin accessibility across the clusters, here's showing the average attack-seq signal with cluster 1 in blue. We see that genes most sensitive to switch SNF inhibition have weaker chromatin accessibility control cells, consistent with these genes being the most dependent on continuous switch SNF activity. And if we look at RNA pol 2 by pro-seq, we see that cluster 1 genes are also more lowly expressed. Next, we looked at histone methylation at H3K4, and we found that cluster 1 promoters have significantly lower H3K4 trimethylation than other genes, which is consistent with our low level of gene transcription. Moreover, the levels of monomethylated H3K4 are elevated in this group of genes. And now this is really interesting because this enrichment of mono versus trimethylation is often considered a hallmark of enhancers. And so thus far, I've shown that switch SNF is critical for maintenance of accessibility at enhancers and at promoters, with chromatin characteristics reminiscent of enhancers. And in data that I don't have time to show you, we found a similar result in human AML and other cancer types, with switch SNF being similarly critical for maintenance of accessibility enhancers and these promoters with chromatin characteristics reminiscent of enhancers, which raises the question, why are these genes so sensitive if switch SNF is clearly acting more broadly? So we next asked if these genes that fail to recover lacked a compensatory remodeler that might be mediating the compensation of the recovering clusters. To do this, we looked at the binding of a number of chromatin factors across our 4 clusters of genes. Remember that it's clusters 2 through 4 that recover and cluster 1 genes that do not. And we found many factors were the same across the clusters, such as the CHG family for remodelers. But 2 factors jumped out, AP400 and TIP60. And these are actually members of the same complex. And they were strongly depleted at cluster 1 genes, the ones that failed to recover. And so this EP400 TIP60 complex is the chromatin remodeler of the acetyltransferase activity. And it's one of the classic coactivated complexes, suggesting that it could indeed be compensating for loss of switch SNF activity. And notably, previous work has shown that this complex is targeted to K4 trimethylation through a PHG finger in its IMG3 subunit, which is certainly borne out by these heat maps of promoters ranked by K4 trimethylation levels. And H3K4 trimethyl is enriched at the recovering promoters in clusters 2 through 4, providing a satisfying explanation for why we see this complex binding those genes, but not those in cluster 1. And so next, what we wanted to do is to test whether EP400 was indeed necessary for the compensation we observe. So as a point of reference, here are meta plots showing the average levels of chromatin accessibility at our cluster 2 genes. We see a modest loss of accessibility at 2 hours and overcompensation observed at later time points. And we'll be looking at switch SNF inhibition for 4 hours comparable to this time point in cells where we have used siRNA to deplete EP400. And I'll first walk you through the results for cluster 2 before showing you the other 3 clusters. ATAC-seq shows that loss of EP400 alone had minimal effects on chromatin accessibility, consistent with previous work. However, when we treated cells with a switch SNF inhibitor, now loss of EP400 strongly reduced promoter chromatin accessibility, indicating that EP400 is necessary for the recovery observed following BRM14 treatment. And the other clusters that recover, 3 and 4, also show this EP400 dependent recovery of chromatin accessibility. However, at cluster 1 genes, which are uniquely sensitive to loss of switch SNF inhibition, we see no effect of EP400 knockdown, consistent with EP400 not being found at these genes and these genes being unable to compensate for loss of switch SNF. Thus, we see that EP400 is necessary for this recovery, and we think that this compensation mechanism explains some of the confusing effects of long-term depletion of switch SNF. And now our data suggests that the EP400 TIP60 complex will have increased importance in disease states where switch SNF is disrupted. So to explore this idea, we used the CBio Cancer Genomics portal to access mutation data for non-small cell lung cancer patients. And here each patient represents a single patient, or each column represents a single patient sample, and plotting the predicted loss of function mutations in each genes and comparing EP400 mutations to 2 most commonly mutated switch SNF subunits, BRG1 and ARID1A, we see a mutually exclusive relationship consistent with cells being increasingly reliant on EP400 when switch SNF function is perturbed. But can we test this more directly? To do that, we took the non-small cell lung cancer cell line A549 and compared wild type cells to those where we had knocked out EP400 using CRISPR and measuring growth after 8 days treatment with increasing concentrations of the inhibitor. And what we see is a dramatic sensitization of the knockout cells to BRM14, providing really nice genetic support for our model that EP400 is important for this compensation following loss of switch SNF activity. As an additional test, we repeated the experiment using a PROTAC degrader of BRG1. And again, we see a strong sensitization of the EP400 knockout cells to loss of switch SNF function. And so with 2 different drugs acting through disparate mechanisms, we observe a strong genetic interaction between switch SNF and EP400. And in data that I don't have time to show you, we observed a similar synthetic lethality across a number of cancer cell lines, spanning multiple cancer types and switch SNF mutational status, suggesting that this really is a general mechanism. And these results, together with the patient mutation data and the molecular results in the mouse embryonic stem cells, we think provide strong support for our model, where EP400 can compensate for loss of switch SNF activity. And so this brings us to our model and our conclusions. And so here's what we found, is that chromatin remodeling is constant and dynamic, and switch SNF acts to open up chromatin and promote transcription at nearly all promoters and enhancers. However, long-term inhibition of switch SNF function reveals differential compensation by EP400. So at enhancers and our cluster 1 genes, these are not bound by EP400, and these are entirely dependent on switch SNF. So when switch SNF is inhibited, we observe a persistent loss of chromatin accessibility and transcription. However, most promoters are also bound by EP400. These are the genes in clusters 2 through 4. So when switch SNF is inhibited, EP400 is able to compensate for the loss of switch SNF. And we think that the synthetic lethality we have observed, that we have described, may open up new doors for treatment of diseases with switch SNF or EP400 TIP60 mutations. And finally, with prolonged switch SNF inhibition, we see a profound disruption of promoter and enhancer communication, with the compensating genes seemingly able to recover despite a sustained and global repression of enhancers, which raises the question of whether the loss of enhancer input underlies some of the perturbation in gene expression in cancers where switch SNF has been inactivated for long periods of time through mutations. And so with that, let me just say a big thank you to everyone in the Edelman lab. It's been a really supportive environment, especially Karen and Eileen, who did so much work on this, as well as Christy, Claudia, and Piero, who also helped. And thank you all for listening, and I'd be happy to take any questions.
Right. Thank you so much for that, Ben. And congratulations on the recent publication.
Thank you.
We're happy to open up the chat now, or if you want to raise your hand and we'll unmute you. Feel free to ask any questions. Go ahead, Jessica.
Hi, thank you for the lovely talk. Earlier on, you mentioned ProSeq, and did I hear incorrectly, or did you say that all NTPs were removed from the cells? And
Yeah,
how is that done?
Yeah. So it's the way it works is you collect the cells and put them on ice to kind of stop transcription in its tracks, and then you permalyze the cells with detergent. And then you wash them, and in that process, all the endogenous NTPs float away. And then what you do is you take those cells where polymerase has been locked in place, and you do essentially an in vitro run-on reaction with the biotin-related NTPs. And because those are bulky, then Pol2 incorporates a single, essentially one nucleotide, but it's got a biotin on it. So you can pull that out with just stripped abdomen. And so what it gives you is the kind of where the active site of polymerase is at kind of base pair resolution on which strand as well.
All right, thank you, Jessica. Go ahead, Matt.
Yeah, that was a really nice talk. Thank you. It's an interesting question. I don't know if it's a silly question, but I was wondering about the order in which things work. So if you were to knock down or inhibit EP400 TIP60, would switch SNF compensate for those genes? Like, or does switch SNF work first and then EP400 comes in after?
Yeah, no, that's a great question, and something we're hoping to get at in the future more incisively. Right now, we've done what other people have done before, which is knocking down EP400, and so for like 48 or 72 hours. And when you look at that time frame of 2 to 3 days, and you do a tax-seq, there's almost no change at promoters. And that other people have seen it before, and that's what we see in the embryonic stem cells. And so our guess is that means that switch SNF is compensating for loss of EP400. And we see that BRG1 is bound at essentially all genes, and then EP400 just at these kind of 75% that recover. And so our guess is that switch SNF is able to compensate for loss of EP400 quite broadly. And it is only when you get rid of both that you see kind of a sustained loss of chromatin accessibility. We'd have to do something like a Degron on EP400 to be able to do the rapid depletion and then see what happens to see if there's an initial repression followed by recovery or not. And that's something we haven't done yet.
Makes sense. Awesome. Thank you.
All right, and we have another question from Michael in the chat. Is EP400 always at clusters 2, 3, and 4, or is it recruited after 2 hours of switch inhibition?
Yeah, that's a great question. So we see it there beforehand at clusters 2, 3, and 4, and then we looked at 2 and 4 hours inhibition and what happens to EP400, and it goes up initially at 2 hours, and then actually comes down a little bit at 4. And so we think that it's being recruited more to, there's an increased recruitment of it to these clusters 2, 3, 4 genes following switch SNF inhibition, but there's still a delay before we see recovery. So we're not quite sure mechanistically what's going on, but we think there's an initial recruitment of EP400 followed by delayed recovery. And we don't know what causes the delay.
Thank you, Michael. And our last question, Gabriel, feel free to unmute.
I thank you for the presentation. Can everybody hear me okay?
Yeah.
Okay. I have a question about kind of transcription itself. So in order for a gene to be expressed, you gotta have both, right? The chromatin accessibility and also the transcription factor. Is there a significant overlap of transcription factor binding, like of transcription factors that actually bind to both of these complexes?
To complexes that bind, to switch SNF and EP400?
That's right, yeah.
That's a great question. And I don't think we know the answer to that. I mean, they're both being recruited to many of the same genes, but whether that's through the same transcription factors or different or like as part of the transcription process can recruit it. Like they both have been reported to bind RNA, for instance, and the yeast form of EP400, that complex, is reported to bind to naked DNA and so be recruited to genes with strong nucleosome depleted regions. My guess is that some transcription factors will overlap in terms of how they recruit and some won't. But I don't think we know enough yet to answer that question.
All right, thank you everyone for some great questions. At this time, we're gonna switch gears to our next presenter. So I'd like to welcome Dr. Martin Schmeing with his talk entitled Structures and Functions of a Non-Ribosomal Peptide Synthase. Take it away.
Thank you very much, Kathy. Thanks to Matt and to the entire organizing body for allowing me to be here and give you a hopefully nice talk about the work that's going on in my lab. So let me share screen. Share the correct desktop and go into the right mode. Is that right? Everybody sees what they think they ought to see? Can't see anybody's
Looks good. Yeah,
looks good.
We're all good.
Thank you very much. Okay, so I'm going to tell you about a story, and I've just seen that my second slide has arrows out of order, but that's okay. The advertisement talked about non-ribosomal peptide synthetases. I'm going to talk to you about non-ribosomal peptide synthetases, so I won't be insulted if you say, that's not what was advertised, I'm going to leave because of that non. That's meant to be a bit of a joke. It's a non-ribosomal peptide. So what do I mean by a non-ribosomal peptide? So obviously, I don't mean a ribosomal peptide. So ribosomal peptides can be the ones that we think about more often, i.e. proteins. They can be these other kinds of classes of small molecules where you make a peptide from the ribosome and you modify it, or the enzymes modify it into a nice small molecule that has drug-like properties. These are called RIPs. And this is why we don't eat Smurf mushrooms, this RIP alpha amidin. So they're not ribosomal peptides, but they're also not non-ribosomal peptides, because non-ribosomal peptides exclusively, or in my mind, exclusively refer to these canonical non-ribosomal peptides that are made by enzymes that are modular and called non-ribosomal peptide synthetases. You know about some of their products: cyclosporin, daptomycin, penicillin, and our favorite is linear grammin shown here, gramicidin shown here. So it's not a ribosomal peptide, it's not a non-ribosomal peptide. Two nons don't make a ribosome. So what is it? It's going to be, the talk is going to be about this non-ribosomal peptide. So this is quite a funky molecule, kind of unwieldy and all over the place, until you start breaking it down as to what it's actually made of. And then you can see that it's actually quite a simple molecule. What you have is a backbone that is all asps, asp, all the way down, aspartic acid. And there's arginines decorating the asp side chain at every single location. And this is the non-ribosomal peptide that I'm going to tell you about its synthesis, its use, etc. today. So we can break it down even simpler by using the N annotation. So it's essentially just a beta dipeptide, asp-arg, through an isopeptide bond, repeating. And it's repeating for a very long number, 80 to 400, which makes it as big a strand of cyanophysin. That's the name of this thing. A strand of cyanophysin is as big as your standard protein. So it can be called beta asparage to the N, or as I just said, cyanophysin. And it's called cyanophysin because it was discovered in cyanobacteria. And it was discovered quite a long time ago. It was named by this old Italian dude, Antonino Borzai. And it was named cyanophysin because it was in these cyanobacteria cells. So using staining and just a light microscope, he could see large bodies of dense things in the middle of these cells. And he was able to guess that it had something to do with being proteinaceous, not that they really knew what that meant back then, but he knew that these were probably important because they were so prominent in these and other cells. And he put forward a couple of papers, including this one, which I actually found a reprint of from 1887. And that ends a diligent and somewhat more detailed study than I myself have been able to do around cyanophycin would certainly be desirable in Italian, not in English. And when an old Italian guy that looks like that makes a statement like that, we think it's a call to arms to figure out what's going on with cyanophycin and how it's made, and some additional information about what it's used for, etc. So what is it used for? Typically, people think about cyanophycin being a fantastic nitrogen storage molecule. You can see all the nitrogen around here, nitrogen, nitrogen, nitrogen. You can see my cursor, or you can't see my cursor?
Yep, we can
see it.
Awesome. So it makes this molecule 24% nitrogen by mass. That is the highest polymer percentage of any of nitrogen in any polymer that we can conceive of or that we know exists in nature. So it makes it a really great nitrogen storage. This makes it very, very useful for lots of different bacteria, but especially in nitrogen fixing bacteria, because photosynthesis is aerobic and nitrogen fixation is anaerobic. So nitrogen fixation will completely stop if there's oxygen around. So those two things have to be separated. And usually they're separated either in a day-night cycle or in a cell-specific type cycle or arrangement. And if you're fixing a lot of nitrogen, they can't go in straight into all the metabolic pathways you want to use them for. You need to store them. So if nitrogen fixation is going at overdrive and you want to have that nice extra store of nitrogen, you can make a lot of cyanophycin during the night. And then when we're doing photosynthesis and we need to use the nitrogen, we can break down cyanophycin and push it into the primary metabolism, allowing the cells to grow nicely. And so it's been shown that, for example, toxic cyanobacteria blooms are really aided by the genes that make cyanophycin and break cyanophycin down. There also are a lot of biotech applications. People got really excited about this as essentially a polymer that could replace some plastics, that it would be biodegradable, that it could be used in nutrition and agriculture. And because of this, they have, over many years, 137 years of research, more or less dormant, but or more or less active, but never dormant. There have been papers from many, many labs looking at, among other things, how to make the most cyanophycin in cells. And that's actually a pretty easy first step. All you have to do is put the one enzyme into E. coli or tobacco or yeast or what have you, and that will make your cyanophycin for you. But if you then metabolically engineer your cell to make it a little bit better at getting all the substrates for cyanophycin synthesis, you can get it to essentially be a sack of cyanophycin. So here is a cell of a metabolically engineered strain, natural producer strain. And you can see that it is essentially just a big sack of cyanophycin with a tiny little bit of cytoplasm left over. And in fact, you can get your growing cell mass to have over 50% by dry mass cyanophycin in it. So it can be produced really, really highly, including by Steinbeckel and all who have been working very hard on this. So what about the production of cyanophycin at a molecular standpoint? How is it actually made? You might not be surprised to see that it is made from aspen aerge, because that's what I told you it was made of. It's quite a simple process, actually, conceptually. You take aspen aerge, and with one enzyme plus a primer, so a little stretch of aspen aerge polymer, and ATP, that one enzyme just goes ahead and makes your long chain massive cyanophycin molecules. Of course, having a storage molecule for nitrogen and also carbon and energy only is useful if you can break it down. And we can break down cyanophycin by a specialized cyanophysinase that just takes water and hydrolyzes the main chain to make these beta asparage dipeptides, which can be hydrolyzed by enzymes, general isobipeptidases. And if I have time, I'll tell you a little bit about a specific one as well. And the last step remaining to describe all of cyanophysin metabolism as we know it is that some bacteria have a second cyanophysin synthetase, which is cyanophysin synthetase 2. And if that bacteria is in the midst of breaking down cyanophysin and then metabolic conditions change, growth conditions change, it can say, oh, hold on, we want to start remaking it all again. And I've got all of these beta asparage dipeptides around. I'm just going to polymerize those directly back into cyanophysin. So that's what cyanophysin synthetase 2 does. So this was known, there were a lot of bioengineering for high production studies. There were some biochemistry studies, but essentially this is not far off the knowledge of how cyanophysin was made and broken down at the time that Itai, a talented graduate student in my lab, started tackling this question. So kind of structurally, this was before AlphaFold or AlphaFold 2, and we had only to be able to look at the sequences to be able to see what it might look like. And we could look and see that cyanophysin synthetase 1, CPHA1, would have 3 domains. One domain is completely unknown in structure or function. It knew there were some alpha helices in there, but that's about it. Then the second domain is related by homology to glutathione synthetase, which makes amide bonds, so that makes sense. And it was known from Steinböckel's work that that's going to be the thing that adds ASP to cyanophysin. Then by homology, we could see that there was a third domain, which is the Murray ligase-like domain, and we're going to call that the M domain, we're going to call that the G domain, and we're going to call that the M-terminal or N domain. So if this is adding ASP, the G domain is, then presumably the M domain is adding ARG because it too is related to a class of enzymes that make amide bonds. So that makes sense too. So we wanted to go from that kind of level of, yeah, this domain probably makes this bond, this domain probably makes this bond. We have no idea what's happening over there, to a precise and detailed full knowledge of how this process works. So to do that, we needed objects of study. We needed to decide which cyanophysin synthetases to work with. And at the time, there had been one study from about 15 years previous showing that despite it being called cyanophysin synthetase, it was not only in cyanobacteria, but there hadn't been a thorough study since the explosion of genomic information. So we went ahead and did that. And we were able to find well over 4,000 cyanophysin synthetase 1 enzymes. And it was really interesting to see how far spread they are in the bacterial kingdom. So here are the cyanobacteria, but there's also all sorts of other clades of bacteria that have it. And in fact, maybe we should be calling it bacteriophysin because cyanophysin is a small minority of the places that you find CPHA1. So what we do often in our lab is if there are multiple homologs of an enzyme that will help us answer the same question, we won't just pick our favorite and start working on it. We'll pick 5 or 10 or 15 of our favorites, clone them out, and go for it. And so that's what we did. We took 14 and had them cloned or synthesized. And then of the 14 that we cloned or synthesized, 11 of them were terrible. They either wouldn't express, or if it would express them, they would just crap out in the tube or something just not nice to work with. But 3 of them were great. This one here, a cyanobacterial one, and 2 gamma proteobacterial ones. And then we were able to express, purify them, and also get really nice characterization biochemically and solve all three structures. A little bit later, a Japanese group, this reference here, was able to also solve a structure of a synthetase from cyanobacteria. We were surprised. We didn't know we had friendly competition in this domain. So we studied these structures and we got them by EM and crystallography. And biochemistry, what was nice here is when you have pure cyanophycin, you can get it just to make cyanophycin itself. When you have pure cyanophycin synthetase, you can just throw in primer, ATP, ARG, and ASP, and off it goes making cyanophycin. And Itai discovered or noticed that this starts to kind of form soluble aggregates and scatters light, just like your bacterial growth scatters light. So our go-to easy assay was just the same as a growth assay for E. coli. We would just see how much scattering we would have over time. And this was super useful for Itai to do kind of pilot experiments. He could just throw the conditions together, the enzymes together, and leave his experiment on the bench for a week, and every day just see whether it was opaque or not to see if there was cyanophycin being made. So we were able to get these three structures, as I mentioned. I'm only going to show you two of them because the third one kind of is derivative in a less good structure. But we had a very nice structure of the gamma proteal bacteria from this one bacteria, and it looked like this. And we had another very nice structure of the cyanobacterial one, which I'll call SU-CPHA1 for the name of the cyanobacteria. And what you can see is, for a structural biologist at least, these look beautiful, symmetrical. There are tetramers, but they build up the tetramers very different ways. And we have, we're able to, I won't go into it, but do a lot of comparisons between the different ways cyanophycin synthetase can build up into its active form. But even though they look so different here, the protomer, the building block, the monomer, shall we say, where you have the three domains, look essentially identical in the two structures. And so we're going to use this one here, the cyanobacterial one, for almost all of the display items in this talk. So we've now zoomed into the protomer, and we can see that it is a three-domain enzyme. And instead of the end domain being like out at the beginning, it is at the beginning, but that beginning is nestled in to the two other domains. So we've got the N domain here, the G domain here, and the M domain there. So the N domain is nestled between the two. And what we could do is solve a bunch of different cryo-EM structures with different substrate analogs, intermediate analogs, et cetera, to see how the active sites both work. And we can just briefly kind of summarize them here. So what we have in this stick figures here is the cyanophycin that's being grown at the G domain, and it's going to get added to right at this position here. What's going to happen is you're going to get phosphorylation, and then you're going to get isopeptide bond formation. So this is the configuration right before the first step of reaction, phosphorylation, and we have a nice structure because that will make a bond with that, and so it's the relevant form. And what we can do is make mutations around the substrate binding site to see indeed that is, these are very important for substrate binding. So that's how the blunt end of a cyanophycin gets one extra ASP added to it. And then if we go over to the M domain, we can again see what that addition will look like. So now instead of a blunt end, we've got a blunt end plus one ASP. And then that means this ASP side chain is going to get added to, again, but first by phosphorylation and then by isopeptide bond formation. The eagle eyes of you in the audience will see that this is a nitrogen and not an oxygen. That's the trick that we played to stop the reaction in this step and not have it keep going further. But this to there is the bond. And so we have this very nice configuration of the active site with a lot of interactions binding the substrate, which is cyanophycin plus an ASP, bound to it to get the ARG bound to the side chain. And in fact, there are so many interactions that most of the time it's redundant. So if you mutate, for example, this glue up here, then you're actually not having very much downed activity. But if you mutate the business end where this arginine is orienting the final asps for addition, then you kill your activity. So this all makes sense as well. So that was really nice, but it actually wasn't that surprising. We got some of the details. For example, I didn't mention, but it was known that this enzyme wouldn't just add asp all in a row and then come back and add the arginine side chains. It would always go asp, arg, asp. And this structure shows why this is. This is clearly intense binding of the full cyanophycin with room for just one asp at the end. So that's why the substrate is bound like this. And that's why you have to alternate adding asp to the backbone and decorating that with arg iteratively. So that was nice, but it wasn't super surprising what was happening there. You would first get asp and then you would get arg added. So then what we wanted to do is look at the endomain and look at what the endomain did itself. And there we would be in for a lot more surprises because the endomain, as I said, is something that didn't have homology to other proteins. It had low sequence conservation, and it had no apparent catalytic importance in any of the reactions that anybody had done or the ones that we had done previously. And the structures also say that it's very far from the active site. So the active sites are up here and up here. What's the deal with this endomain that's always there in cyanophysin synthetase? What's it doing? So the answer, which we've got to in more than one slide, but I don't have time to tell you how we got there completely, is that it binds cyanophysin as well. It binds the polymer as well. Because when you're doing an EM structure and you have polymer in there, but not when you don't have polymer in there, you see extra density, as you would expect. You see extra density over here. This is the active site of the G domain. You see extra density over here. That's the active site of the M domain. But you also see extra density here. And that is hovering over the N domain. So it's over this area of the N domain. And what we found was these two helices that line up next to each other and point up towards the active sites are extremely charged. One of them is very, very negative, and the other is very, very positive. And in fact, I said there wasn't conservation, and that's true. But if you take just a family, a clade of bacteria, then these are very well conserved. So for example, in cyanobacteria, helix 1 is very highly positive and helix 2 is very highly negative. Whereas in Gameoproteobacteria, it's flipped. Helix 1 is negative and helix 2 is positive. So that's why you've lost the conservation information. But if you mutate these helices, if you take out the positive charges of one, or if you take out the positive charges of the other, or both, then you get progressively lower and lower activity, which again is remarkable considering how far away from the active sites they both are. And what's nice is if you add back the charges by swapping the negative charge over to the helix 1 and the positive charge over to helix 2, then you recover back information. So essentially what this means is that we've got a track of charging that serves as a very nice loose tethering mechanism for cyanophysin. So if I play this little video, you can see that that allows cyanophysin itself to stick close to the active sites and get fed in to one active site where, as I showed you the details, asp will get elongated, or the chain will get elongated by putting on an asp, and then that allows it to track over again to the ARG active site adder in the M domain. So in the last 5 minutes, I'm going to tell you about another job that the end domain does, which was completely surprising and novel. So I mentioned several times throughout the talk that there is primer required for synthesis, so that you need a little bit of asparage to polymer to start off your synthesis well. And that's true typically, but what Itai found was where that's strictly true in the Gameoproteobacterial one here, TMCPHA1, you can leave the reaction conditions without polymer, and for hours and days, you'll get no polymer formation. You'll get no cyanophysin formation. It wasn't strictly true for cyanophysin synthetases from cyanobacteria. So with those guys, there's just a delay. There's a lag of 15 minutes. And then like clockwork, after 15 minutes, boom, it starts making cyanophysin. So with polymer, it would go polymer or primer added to the reaction. It would go straight up. Without, there's a 15-minute delay, and then it goes up. So what's going on with that? So I'm going to call this one the primer independent CPHA1, and this one the primer independent. Pardon me. This is the independent, and that's the dependent, as it says on the slide. So Itai spent 3 years looking at the 2 active sites, because this was a great setup. We had a primer independent structure, high resolution. We had a primer dependent structure, high resolution. Surely we could look at them and see what was different about the active sites, because the most likely explanation is one or the other one can bind the very small substrates, which would be asp on its own, or a single beta asp arg dipeptide, would bind those better. And that's why you get synthesis more in the primer independent one. So he spent 3 years and dozens of mutational work data that couldn't figure it out. Nothing seemed to be really substantially different between the active sites in the G domain and M domain of primer dependent versus primer independent. So that left only one domain, which was surprising. And I remember talking to Itai in the office, and we were saying, well, this can't be, because there are only 2 active sites and you've analyzed both active sites. The only possible explanation is there's a 3rd active site, but that can't be true. And of course, it was true. So if you take the back of the end domain and look at a conserved area, Itai was also working on proteases or cyanophycinases at the time, and he recognized that this back of the end domain looked a lot like a zinc protease. And in fact, if you mutate any of these residues and don't produce primer, supply primer, then your primer independent synthesis absolutely tanks. Mutation of these residues don't do anything when you do supply primer, but when you don't, it tanks. So that means this is completely necessary for this primer independent synthesis. And in the end, what it is that it will be able to take a bit of cyanophysin polymer that already exists and break it down by proteolyzing it into primers of 4 asparage dipeptides in a row. So that was extremely super surprising, but awesome to find an active site in an enzyme that had been studied or known about for very, very many years, but no one expected an active site there. So that was cool. And what the deal is you can, both the independent and dependent enzymes can make very, very, very slow amounts of cyanophysin, but only the ones with the active site back here to chop them up can then take that very, very small amount, chop them into primers, and make large amounts. And that's what we're seeing in our assays. Okay, so with that, I'm going to end with 2 things. One, I'll say that Itai was awesome, and he solved the structures not just of CPHA1, but also of every other enzyme in this pathway, and really crushed the field, or destroyed the field, as his thesis examiner said. And also, I'm going to end with this overall movie. You've seen a clip from it, but I'm going to show the whole movie now, which shows kind of synthesized the way we think that cyanophycin is made. I'm not showing the cleavage because that was a second paper, and this movie took me 3 weeks to make, and I didn't go back and spend another 3 weeks on this. And as the organizers know, this is a musical movie because there's a tradition in my lab to make musical movies with nerdy dad jokes in the music. For example, the first music is Welcome to the Machine, because this is a macromolecular machine. And then it goes on and is just as corny about elongating things or adding things and decorating side chains. So here we go with the musical movie.
Welcome, my son. Welcome to the machine.
Kind of bison sliding up. It's built up. That carbon is going to get added by phosphorylation and peptide non-peptide. It's going to slide. Over to the
Glide, and let your backbone slide.
back it goes over to the activation. Over to
And let your backbone slide. I love when I slide.
adding, sliding,
Separate my side.
really good depth and build up a long chain.
Don't you remember?
All of the different active sites can be firing at the same time. We build up these
City on rock and roll. We built this city on rock and roll.
That's the deal with how cyanophycin gets made, and these are the people involved. Itai was the main hero here. He did an absolutely fantastic job, and he was helped by first by Asriel and then Linda. And it was a collaboration with Don and Marcus to get the small molecules. And I did my sabbatical with with Andreas at UCSD to get the first structure. Yeah, if there's time, I'm happy to take questions.
All right. Thank you so much, Martin. We're going to do one question for this round, but everyone is more than welcome to connect with Martin offline. So our first question here in the chat from Mike Downey: how processive is the enzyme? Is it one continuous run or binding / release cycles at active site? What impacts such dynamics?
Yeah, that's a great question. And so there are several phases to the answer there. And what we know, that's not quite the right answer, the thing, so I won't show that. So what we know is we have these two active sites, and when you have got the really small molecules, so asparage 4 primer or something shorter, then those have to disengage and then float away and re-engage later. And we know that that's a bit of a slower process, but once it starts getting built up and can start to get tethered to the end domain, loose tethering to the end domain, that's when you're about 12 mer or more. And then we think it's more common for it to be the same molecule that's going back and forth between the active sites. So they're definitely different phases, but once it gets going and is working on a longer molecule, everything indicates it's really quite processive.
All right. So thank you so much to everyone for coming to this week's seminar. A big thank you to our speakers today. They both gave some really fantastic talks. At this time, we'd like to invite everyone to join us in the new year in January for our next installment of the CSMB seminar series. And with that, we hope you all have a great day.
Thank you.
Thanks, everyone.
Thank you.
All right. Bye, everyone.
Bye, everyone. Thank you so much for coming.
Awesome talks. Thank you.